graduates with that. We'll be doing some information sessions on those and how you can enroll in that program in the next few weeks. So we've got lots going on in the next few weeks. So check out the calendar on the CURS website. Make sure you don't miss anything. We'll continue to do these research toolkits about once a month. And so if there are topics that you think would be helpful, whether you as faculty, staff, students, anybody, if you have topics that you would like to suggest, we would love to hear your suggestions. So, so please let us know. So just so you know, the recording has started for this. You should have a notice on your screen there. But let me go ahead and introduce Dr. Browning, our guest speaker for today's toolkit series. So Dr. Browning is a professor at MCG and is also part of the Georgia Cancer Center doing research on colorectal cancer and inflammatory disease, and I'm not sure what all other gastrointestinal diseases. I know that's not the topic of your talk today, but that might work its way in a little bit here. And the reason we've asked Dr. Browning to come is because he is a co-program director for the 3MT competition, which is essentially an elevator pitch competition for graduate students. And so, you know, having students learn how, practice, and get better and better at describing their research in a very short amount of time. So the idea of the elevator pitch is, of course, how much, you know, you can get across in a succinct manner and about as much time as would take you to go a couple floors. And so this is used often as a great interview strategy, but it's a great strategy to be able to apply in lots of different situations, whether that's interviewing or research or just kind of what's going on in your world. So, and a professional conversation. So let me, with that very short introduction, go ahead and turn it over to you, Dr. Browning, and you're going to tell us about how to make the most of those couple minutes that you have to describe what you've been working on. And I think you're muted.
Yeah, I am. That's not the first time today that's happened. I had a question before I even begin. So the recording that you're doing right now, that is going to be available so I can actually send students to it. Is that correct?
Sure. Yes, absolutely.
Okay, so let's see then if this is the right screen. So you can see my screen?
Yes, we can now.
Okay, thank you. I have 3 monitors hooked up to this computer, so it gets confusing.
Gotta make sure you get the right one.
Okay, so thank you very much, Dr. Davis and the CURS program for inviting me today. It's actually an honor to present some information that is not typical for me to do. You already went over my credentials. You know, my name is Darren Browning. I am a professor in biochemistry, molecular biology. I've been the graduate program director for the PhD program in biochemistry, cancer biology for over 15 years. And I am a member of the Georgia Cancer Center, where I have a research laboratory focusing on colon cancer prevention. And while I'm not going to be talking directly about it, I will be using some of my research as an example of how to sort of reformat that for more of a lay consumption. But none of these credentials really have helped me learn communication. Really, they've been a hindrance more than anything else. I have other activities that have really contributed more to why I'm here today. And the first one is that I'm a parent. So I have 4 kids. And when your 6-year-old comes to you and asks, you know, where do babies come from, they're certainly not interested in an hour-long lecture about myogenesis or meiosis or gametogenesis. They're interested in where their baby brother came from. So that was definitely, being a parent can help you translate more complicated phenomena into more simple terms. I'm also co-founder and CEO of Target Biotechnologies, which is a company, a hobby company, And so, when pitching your research to venture capitalists, they don't want to know any of the details about your experiments. They're more interested in whether you're going to generate a return on their investment. Lastly, but certainly not least, as Dr. Davis mentioned, I co-direct the Augusta University 3-minute thesis competition. And for those of you that aren't familiar with it, this thing started about 12 years ago at University of Queensland in Australia. And the idea was to get their PhD students across all of the different disciplines to be able to explain in an effective and compelling way what it is that they've been doing for their research. So this is like a 5-year PhD program, and explain it in only 3 minutes or less. And so the ability to do that really was recognized immediately, and it's grown over the last decade to probably this year, it's got to be around 1,000 institutions across the planet who hold annual competitions, and there's also regional, national, and even international competitions. So I wanted to give you an example of how we can use the process of a 3-minute thesis to translate more complicated scientific phenomenon into something that's more digestible for a lay or at least a non-specialist audience. So if you'll indulge me for a minute, actually probably more like 2 minutes, this is some data that was generated by a student of mine a couple of years ago as part of her thesis. And I'd like to very briefly just go over it as I would for a peer audience. So if I were going to give a seminar or I'm talking to somebody over coffee that's my colleague about the research, and right after that, I'd like to show you the presentation that my student did who did the work as a 3-minute thesis presentation. And so as the title suggests of this slide, the goal of this project is to use phosphodiesterase 5 inhibitors that increase cyclic GMP. Most of you know them as erectile dysfunction drugs such as sildenafil, and we show here that they can inhibit carcinogenesis in mouse models of colorectal cancer. And so we use 2 models. Shown in the upper left here is an inflammatory carcinogenesis model that really represents a human high-risk cohort that have IBD. And in this one, we do an IP injection of isoxymethane, which is a mutagen. And this is followed by several cycles of dextrans sulfate sodium to basically destroy the luminal epithelium in the colon, and that gives rise to inflammation. So when we do this, after 12 weeks, you can see here in the distal colorectal regions, there's a lot of tumors. But strikingly, if we give them sildenafil in their drinking water throughout this whole process, we have dramatically less tumors. And this is shown as tumor multiplicity, or polyps per mouse on the right here, where the control gets around 10 tumors per mouse, but we cut that in half when we give them sildenafil in the drinking water. And one of the neat things about this model is that we can get some mechanistic information. So if we give the drug throughout the inflammation cycle shown here, there's absolutely no effect. So we're not affecting tumor promotion so much as if we give it to them early, just during the mutagenesis phase, we get almost the same effect as it has throughout the whole process, which means we're preventing tumor initiation, and we're not affecting promotion or inflammation. So we use another model shown here, and this is the APC-Min for multiple intestinal neoplasia model. And this has a mutation that's common with human FAP patients. These are familial adenomatous polyposis patients, and they typically get hundreds of tumors in their colon. The same mutation in a mouse gives you multiple mutations in the small bowel, as shown here. And again, if we can treat them with sildenafil, we get a dramatically less number of polyps per mouse. And if we use another cyclic GMP elevating agent, this is a guanyl cyclase activator, we get the exact same effect shown quantitatively over here. And in both models, we can cut the tumors per mouse or multiplicity in half. And I could go on about this part of it, but I think I've made my point. I would be surprised if the people in the audience and students looking at this in the future, unless they're studying this field, won't be able to get much out of it. So this title, whoops, I didn't want to do that. Okay, so this title really should be, something that's more appropriate for the audience. So just bear with me for another 3 minutes, and I'll show you how my student reformatted this or repackaged it for a non-specialist audience.
Darren, we don't have any sound. Can you try to reshare that?
Oh, is it not working?
We can see the video, but we do not have sound.
Oh, okay. I talked to you about that. Let me try that again. I forgot to click that button, I guess. Let's see here. Okay.
That sounds like it's going to work.
It'll work if I can just get back to this. All right, sorry about that. 3.5 minutes. Now you can hear it?
Yep.
I'm sure there's not a person in this room who hasn't heard of the wonders of the little blue pill shown behind me. You may have seen commercials telling men that when the moment is right, they may want to ask their doctors about Viagra. Well, have you thought about the women? My research aims to show that this little blue pill may truly be a wonder for both men and women because we believe that Viagra may be able to prevent colon cancer. Colon cancer is the second deadliest cancer in the United States. In 2017, it is estimated that over 50,000 people will die of this disease. That's nearly a quarter of the population of Augusta. What many of us may not realize is that our lifetime risk of getting colon cancer is about 1 in 20 for both men and women. So for every 20 people in this room, one of us may have to face colon cancer in our lifetime. These odds could be worse. Perhaps if a mother, a father, maybe a brother or sister was diagnosed with colon cancer. People who suffer from inflammation in their intestines, such as Crohn's disease and colitis, have an even higher risk. And unfortunately, there are those among us that are born with faulty genes. And for them, the question isn't, will I get colon cancer? It's when. Currently, your doctor's toolbox has very few treatments for colon cancer, which is why the best way for us to fight against this disease is through prevention. So our laboratory has found that when we give mice Viagra in their drinking water, it increases a special chemical in their colons called cyclic GMP. And we believe that this changes the colon in a way that protects it against inflammation and cancer. So we have two models. We have one mouse that experiences chronic inflammation and another mouse that has faulty genes. These two mice mimic people in our population that are at higher risk of getting colon cancer. My results show that when we give these mice Viagra in their drinking water, it reduces the number of tumors by 50%. So while we don't know exactly how Viagra is doing this, our laboratory hopes that one day we can offer yet another drug in your doctor's toolbox to help fight and prevent against colon cancer, particularly in high-risk patients. So now that I've told you that this drug that we're normally accustomed to treating erectile dysfunction in men may actually be a way to prevent colon cancer in men and women, when the moment is right, we may all be asking our doctors about Viagra. Thank you.
Okay, so that presentation by now Dr. Bianca Islam a couple of years ago took the first prize and People's Choice Award for the program. And shown on my left here, or my right on the left here, is Sarah Sharman. She took second place. She was also a student of mine, and she is now actually doing 3-minute thesis full time. So she's a science writer. She just started this position a few months ago, and her job is to go around to the individual investigators at the Hudson Alpha Institute of Biotechnology in Alabama and convert it into a format that's more easily digestible by a lay audience. And so she's the equivalent of Tony Baker that we have at MCG, who actually wrote up the story for this presentation. And this actual story went viral a couple of years ago. I don't know if it's so much the work or just you put the word Viagra into a title and people take a second glance at it. However, so today I'm not my goal is not to try and teach you how to do a presentation that's obviously orchestrated and practiced like this 3-minute thesis, but the principles in learning how to take the complex data and explain it in a way like the 3-minute thesis works in many different circumstances, from just dinner table to elevator, as it were. And so I have 7 different take-home points here that I found are very important over the years. And I'm going to focus most of today's on just the first 4, and then we'll finish up maybe at the end, which is really telling your story before the time runs out today. And so most people might actually gloss over this, but this stuff is very, very important. So I like to take one of them at a time. So the first thing that people need to understand before they even start to think about presenting their data is what the purpose of doing a talk is. And it may seem inherently obvious to you. It's that the goal is to help your audience to understand what it is that you're doing in your research, and then why it's important, but not why it's important to you, why it's important to them in your audience. And so the hallmark of any good presenter is to make something that's really complex and make it seem simple. And so again, this may be inherently intuitive, but I can't tell you how many, not just students, but seasoned faculty have come and given a seminar, or even at a meeting, they're talking to you about their research, and they completely miss this boat. They have different goals, such as to try and teach you everything about their field so that you become an expert, which is just not necessary and diffuses their main point. Or more ego-driven things. They want to show you how smart they are or how productive they've been. And I can give you a little example of that. I mean, this may be a little bit hyperbole, but this is an example of a slide that just has way too much information. And of course, here I am doing a slideshow, but this is the same thing pictorially as you might expect for a verbal presentation as well. Meaning I can talk to you about this particular figure here is human data showing some expression of PKG, and here's some mouse data, which is cool because it's a knockout mouse and we show they have more tumors. We can grow epithelium from the lining of the intestine as organoids. We also show expression data in there, but the cool thing is the knockout organoids grow faster. And so I can go through it. All this data is very important for me, but to me, it's very important, but it's not really important for this talk if you don't understand what I'm talking about. And so the nice thing about having virtual seminars these days is when I see a slide like this, I turn off my video, turn off my microphone, and I go about my day doing other things. This kind of title should really be a completely useless display of productivity. And so for an audience like we have here today, and presumably in future, this makes much more sense. So this is the same information, the same take-home message, but in a pictorial. And again, it can be verbal as well. And the take-home message here is very clear. I think everybody should understand it. Drugs such as Viagra use this enzyme called PKG2 to inhibit or prevent colorectal cancer. Now, depending on the audience I have, I can obviously throw in some more details to say that Viagra can increase the levels of cyclic GMP, and that activates type 2 cyclic GMP-dependent protein kinase. That leads to inhibition of proliferation, and that's what prevents colorectal cancer. But the take-home message, whether you're doing it pictorially like this or verbally, should be as shown here. Okay, so once you understand the purpose of your talk, to make things simple so your audience understands what you're doing, the next thing that I can't overstate enough is to know your audience. And so I have, for some of the research in my lab, maybe 150 slides. For any given talk, I might show 30 or 40 of them, and which ones I choose to show depends on the audience, what they know about your research. Do they have a STEM background? Are they in my field or not? And also what part of my research affects them or would my audience find interesting. So these are very important things to know. And actually, while a lot of people don't take this into consideration when they're describing their research, ironically, you do take it into account on a daily basis. And if I could get maybe an opportunity to demonstrate this, Dr. Davis, can you hear me okay?
I can, yes.
Can you just be sort of a volunteer? And I hope this works. So what I want to do is demonstrate how we selectively describe our stories or our research based on the audience. And we do this really without thinking of it. So I'm not going to ask you to talk about research, but just assume that this morning you got up in the morning and you woke up with a splitting headache, and we're living in a COVID world. So of course you're scared and kind of bummed out about that. And so your response to getting this headache is to take some aspirin or some ibuprofen and cheer yourself up by making a nice big plate of pancakes. You cover them with strawberries and whipped cream, your favorite. And then after that one, maybe you play with the dog, take the dog out for a walk before coming back and getting ready for work. On getting in your car on your way to work, some son of a gun with a huge truck cuts you off, causing you to almost smash into the back of their truck, and then you see them with making some obscene gesture out of their window. So of course this infuriates you, and I'll leave that story there. But you end up getting to work and basically doing the same things that you do every day. So if you can sort of basically get the idea of that hypothetical day you might have. So now what if later on you're at a meeting with your friends, a dinner table. Obviously, this display is pre-COVID, but this is a group of your friends, and you're all sitting around the table, and then your friend across the table asks you about how your day went. So what are you gonna, what kind of story are you gonna tell them?
I'm going to start with that terrible story at the very beginning of my day. Probably not the mundane stuff that is typical of my day.
So more like the almost car crash and
Yes.
how crazy it was, and you slammed on your brake. So you're going to basically try and come up with some information or some story that's going to be interesting or entertaining or captivating for the audience that you have. So that's certainly right. So now let's change the scenario a little bit. So now you're at your doctor's appointment, and yes, this guy's really looking at you creepy, but if he says then, so how was your day? What are you going to tell him?
It was just fine, thank you very much.
So at least you might say, well, I had a splitting headache. I had the pancakes. So I'm not eating well as I should be. I'm kind of bummed about COVID, whatever. You're going to basically tell things that you think he might be interested in. So let's switch it up again to this group of children. And so one of these children asks how your day was.
I would probably say, I had some really good things and some not so good things, and probably want to turn it back to them because that's what they're going to be interested in themselves in their day. Maybe.
If it was me, you're looking at the kids, and I'd probably say something like, well, I had a great big plate of pancakes. Does anyone like pancakes? And played with my puppy, or this, that, the other. So basically, it's a context-sensitive answer that you would have. And so this is the take-home message from this non-scientific sort of experiment we just did is that really what you say with the details and how you talk about it really depends on your audience's knowledge level and their worldview. And so that's a very important point. So if we translate that now out of this sort of day-to-day thing and now translate it into your research. So most of us in academia, this is actually a slide that I took from a seminar sometime, again, pre-COVID, but we're trained to, give a presentation in front of a podium with a pre-made slide deck showing all of your information, and you've got a captive audience of people that are educated, and they chose to come to the seminar to hear about what you're talking about. But this is this is one audience, and it's not the only kinds you'll get. So if you're thinking about describing your research to this group, how would it change? Or just think about how it would change if you now had this audience. You still have a podium and a slide deck, but now if you look at this audience, these are not specialists. They didn't come here probably to hear you specifically. If you look at, I don't know how big your screen is, but if you look at this woman, she's probably thinking about whether she let the dog out and is going to pee all over her carpet. This guy's wondering whether he can hold his bladder throughout the whole talk, and this guy's falling asleep. So what are you going to say about your research that's going to captivate this audience? So how is your research affecting them in a way that's going to draw their attention to have them listen to you describe your work afterwards? So here's another group. And obviously, this is a bunch of children again, so how you're going to explain your research to them has to be in a context that's going to be meaningful to them. And I've been in this kind of situation, having two daughters, and bring your daddy to work day or what have you. And so it can be very challenging. So I'm not going to talk about the things we do with mice because, these young children may have rodents as pets. And, talking about cancer can be a very sensitive topic as well. And Viagra, I wouldn't even go there. So, the point is you have to sometimes consider your audience and pre-plan what exactly it is you're going to talk about. And so here's another one, just a couple more examples. So this could be, venture capitalists looking to hear about your research, or think of it as for some of the students, maybe it's a job interview, and these people are asking you about your research. So they really want to know the very specific details and some of the data, or do they want to know what you learned from it and how they can, you could advantage their company, by that learning process. So again, different audience, different story, and like that. So this is the final one, and the only one that's actually context sensitive. So you're flying somewhere for a vacation maybe, and this is your seat. You can't change seats, so you sit down next to this guy, and he asks you, so tell me about your research. And so if I were to ask you, what are you going to say? The correct answer in this case is you can't really say anything. You have to find out what it is that this guy knows. Is he, he could be a history teacher in high school. He could be an architect. He could be a janitor, or he could be a physics professor. You don't know what he is, so you don't know the knowledge level or his worldview. So it makes it very challenging for you to describe your research to him until you know a little bit more about it. And so, very often, if I don't know who they are, I will ask them first, what their interest is and what their background is before going on with my work. And I can example that as well on some of my research that I've shown you already. So this is a simplified version of one of my projects. Erectile dysfunction drugs can inhibit colorectal cancer, but it's just a very small part. So it makes sense if I show this to you now or verbally say these things, because there's a non-specialist audience. But if I were to be presenting this research in front of, investors for my company, for example, I would tell them that, there's a whole family of these drugs, and we're synthesizing our own drugs that are unique to the digestive tract, and so they're interested in the potential market for these drugs going forward. So I would spend a lot more time talking about this and how it can relate to that in a market sense. But we also in the lab are targeting these drugs for chronic constipation, other functional bowel diseases, as well as leaky gut and this associated diseases with that. So if I were presenting this research to physicians, I might spend less time on this and a lot more time talking about how maybe these drugs could help their patients with these kinds of things. And again, if I'm in front of a more academic audience that is more in my field, I might talk a little bit more about some of the animal models or depending again on the specialty of the audience, about some of the mechanisms that we think underlie all these. So there's one story, and you choose what you think is going to be the best or the most impactful given the audience that you have. All right, so once you know why you're talking, to convince your audience that what you're doing is important and explain it to them in a coherent way, and you know who your audience is, the next thing that is surprisingly problematic for students, PhD students, to get in front of is really understanding their story. Because typically students are doing many things, even the undergraduate students in the lab, the ones in my lab, they're doing more than one technique. They're doing many different kinds of experiments. So identifying the main goal of their work is a challenge. And once they've done that, then they have to figure out how to tell the audience that it's important to them. And so I want to focus a little bit more time on that one. And in order to understand why this can be a challenge, scientists, and this doesn't necessarily have to be like a STEM scientist. This can be anybody that is an expert in their field trying to explain their work to non-experts in the field. And so keeping with the research as a scientist in a lab, you're in a day-to-day basis, you're occupied with very detailed experiments and setting things up, trying to get them to work, generating data, and then analyzing the data. And this is your daily grind. And so we call them basically being in the weeds. most scientists, definitely, I would say almost all students live in the weeds, and they have a challenging time even just getting up to the trees so they can see the weeds, meaning can they even see the field-specific point of their research. And so the problem is that if you're speaking to a non-specialist audience, very smart people, like I said, if I'm doing biomedical research, you know, an architect or electrical engineer, very smart people, but they're not going to understand anything about the weeds. So if I want, they're basically up here, they're in the sunlight, they don't know anything about this. So my job is to try and get them to understand what I do. I have to basically show them the impact of my work on them. And so I term this the big picture. And some people say, is that really a useful thing to do? But if you think about it, if we look at actual the population size, the specialists, the scientists are really a minority. And it's the public or the general, the more general non-specialist audiences. There's so many more of them. And typically, I mean, these are our government officials, these are taxpayers. So they're the ones that actually fund your research. So we really are obligated to have the ability to explain to them what we're doing and why it's important to them as a whole. And so this being in the weeds and not being able to see your research from this greater big picture is one of the things that students have a hard time with. So what's a piece of advice then for a student trying to identify what the main goal or the big picture of their research is? And basically, you have to sort the signal from the noise. So what does that mean? It means out of all the things that you're doing on a day-to-day basis, you have to try and get your head out of the weeds and ask yourself a couple of questions. So the longer term or the big picture impact of your research, that's more easily found if you said if you're successful, and anyone that follows you, the lab that you're working in as a whole, if they're successful in the long run, who is it that's going to benefit from that? That's a very important question. That's sort of taking you from the weeds. If you're successful over many, many years, then who's going to actually benefit from reap the rewards of your research? Another way to look at it is, what is the current state of affairs that you're actually trying to improve upon? What's the current situation that's wrong? What's wrong with it that your research is going to try and fix? And so asking these two questions should give you some idea about what the longer term impact of your research is. And so again, this is a challenge, and I would recommend that all undergraduate students make a point of working with their mentors to find out what that big picture is. And it wouldn't surprise me at all if even some of the mentors that are guiding those students also have to actually do a little bit of research and a lot of introspection to figure out what the long-term effects of their research are. I know because even myself, I had to sit down and think, okay, what cohort of patients am I really trying to advantage by the research that I'm doing? And so this slide is actually, this is what I do in the 3MT, is try and help the student begrudgingly come up and see their research from a slightly higher vantage point. All right, so once you have figured out what the larger, bigger picture impact of your research is, it's important to be able to say something to try and convince your audience that your research is important to them. And this is also equally challenging. And again, without having a whole group of students to serve as examples, and this is also very challenging in a virtual environment to do. It's much easier to just pick somebody out and say, okay, tell me about your research and we can use an example of it. But there are some certain techniques that we found during the 3MT competition every year that are quite effective, and I'm listing them here. And so if you understand what the importance of your research, meaning the big picture, how do you convince an audience this is important to them? So oftentimes somebody will come in and say, well, I'm studying ovarian cancer, but then, you know, maybe a lot of the people in the audience don't really care. They're not going to get ovarian cancer, they're men, or they don't know anybody. So that means that half of your audience might be turned off already and they're not excited about it. But if you can bring in something that is going to say, you know, people or animals or children are suffering or dying or they're being harmed in some way, you can invoke the passion. So ovarian cancer is an example of something that you're trying to find a cure for. There's people that are suffering, and those people could be somebody's mother or somebody's daughter or somebody's sister or something like that, and then bring in some statistics maybe about how important it is, in terms of a healthcare burden, something like that. And so these are some of the tricks. Either something like I've described here. Again, I'm kind of tainted being biomedical, but this could be, you're studying pollution or river control or forest control. How does that impact the greater population? There's always a reason, or there's always some kind of a reason that you can come up with, and the basic situation where somebody is being harmed by the current situation, and your desire is to fix that, is one good way to do it. Another one is to invoke fear for diseases, or for toxins in our air or our water or something like that, whatever the research is. Then telling the audience, again, giving statistics potentially that shows that they're actually susceptible to these things, can evoke some kind of a fear, so they want to know what you did to save themselves or their loved ones, depending on what you're doing again. And then lastly, again, if you're not invoking compassion or fear, then the current situation can be an unacceptable societal cost. Again, this could be just pollution or traffic, or it can be diseases, taxes. There's a healthcare burden that costs us. And so again, there's always a way to make the larger story that you've discovered that your research impacts into something that's compelling to that specific audience. But this is something that you need to do and check off that box before you really start talking about your research to begin with. All right, so once you've done that and you've got your audience captivated, and then they want to hear what you have to say about this topic now, there's a new challenge, and this is basically telling them your story. And I've broken this up into 3 different parts that, again, we found to be effective. If you eliminate any one of these, then your story is incomplete and your audience has question marks. So the first thing you do is tell them your idea. And then the next, you tell them how you're testing the idea or implementing it. And then lastly, you tell them what you found. So this is basically telling your story. And there's multiple components to that. Once you've basically identified the big picture impact for your audience of your research, then you basically describe your hypothesis, and you never lose sight of that big picture. How does the question you're asking affect the big picture? Then how are you testing your hypothesis? This is when you find the cool experiments, but mostly the key experiments that are going to describe your story. You can't explain everything to them, just a couple of them that are going to show them what you're doing in your research, so they have some understanding of what you did. And then finally, you show them either some of the results that you found, or what you expect to find, as research projects are typically ongoing and you have expectations. You don't always have all of the data on hand. And so this is another diagram using my pyramid, just to show, again, the audience is up here. They don't know that much, and this is you down here in the weeds. And so how far up these lines are basically describe the level at which you're supposed to be talking to the audience. So right in the very beginning, you identify how your research affects the audience, the big picture. And usually the hypothesis or the idea that you have is much further down into the weeds. And so in order to just get them to understand what the picture is and how or your hypothesis is and how it relates to the big picture, you need to educate them. You need to teach them something that will allow you to focus in on this smaller component of the big picture. And then finally, in order to describe some of the experiments that you're doing in your research, then you need to educate them further. So this is going to be how a particular piece of equipment works, or whatever model that you're using to test your idea. You need to educate them again. So this is at least 2 times that you need to use some tricks to educate your audience. So once you've done that, again, the experiments must relate directly to the hypothesis. The hypothesis must relate directly to the big picture. You can't just lose it and you just get them lost in the weeds. You always make sure that it's contiguous. And so when you're describing your experiments, then you're going to talk about the kind of results. That's about as far down into this pyramid as you're going to get. When you're talking about the data that you've obtained, you don't want to bring them any further down into the details or the weeds other than that. Once you've got your results, then the last part of this is to bring them all the way back home again and say, well, how do these results, what has to happen with these results to generate some effect on the big picture, which is basically, so what needs to happen next in order to change the world? And so you sort of start them off on their level, you bring them down into your world a little bit, and then you bring them back to how your research is affecting their world. And so we found this to be very, very effective. And if you did care to look at a bunch of 3-minute theses presentations online, you'll find that this is pretty much the typical format that people take. So if I take you back in the last couple of minutes now to this really crowded slide, if I was going to use this to describe this to you in a short period of time, I have to look at this and decide what's the most important experiments to get the idea across. You can't show everything. It's going to put people asleep. They're going to be tired before you even get to figure 3. So you have to identify what's the key experiments, and then you need to educate them on, for example, the techniques that are used, and then explain the data that you got on just a couple of them. So for this particular crowded slide, I might choose to look at this one because it's pretty cool with the knockout animal and the tumor model. Growing organoids is really neat. So I'll just choose a few of them, and you pull them out and you focus on those. Now, all those other experiments that I showed are very important if you're a scientist, to cover all of your bases, but they're not necessary to make the key point. And if you're doing research, then typically there are key experiments that you do that really prove your hypothesis, and then the rest of the experiments are usually icing on the cake, as it were, to round things out. So these are examples of key experiments that I would choose to describe that research. And the first one is, again, you have to educate people. So this is a technique known as Western blotting that allows us to see this enzyme known as PKG2. And then in the mouse, we use genetic tools to actually delete the gene that codes for PKG2. So in these mice, this is showing 3 normal mice and 3 mouse that we've actually knocked out the gene for PKG2. And when we look at the colon cancer in these mice, this is that inflammatory carcinogenesis one. We see compared to the normal animals, the ones that don't have the PKG2 get almost twice the number of tumors per mouse. So this really suggests then that this PKG2 enzyme is somehow inhibiting the formation of tumors or prevents colon cancer. And so we think that this PKG2 does it by inhibiting the proliferation of the cells that line your gut. That's where the tumors come from. And so we're able to take from the normal mouse, we're able to take some of the cells that line the colon, and we're able to grow them in a test tube in the laboratory. And the faster they grow, the bigger these organoids they become. And so we can just measure them. And that's what we show here. So this is over time in culture, the size of the organoids. And this is the ones that contain PKG2. This is the ones where we've knocked the gene out. And you can see clearly if we knock the gene out, then they're growing much faster. And so basically, here's 2 experiments that demonstrate that PKG2 can suppress colon cancer by inhibiting the growth of the cells that actually form the cancer in the first place. That is it would take me hours to go through each one of these figures, but that's enough to basically explain the whole research line that I wanted to get across. All right, so I'm going to finish up, actually. This is my second to last slide with how do you teach a non-specialist audience the important aspects of your research. How do you get them to understand your hypothesis? And then once you've got them to understand your hypothesis, how do you get them to understand your experiments so that they can get a grasp of the kind of results that you get? And so I've come up with just a few rules that are critical to be successful at this. The first one and foremost is to get rid of all technical jargon. Anything that is not used in everyday language, if it's not essential, then get rid of it. If it's really essential for them to understand this term is the only thing that describes the phenomenon, then you have to tell them about, explain what that term means. But by all means, get rid of anything that is not commonly used in everyday language by non-specialists, and that's going to make things much more understandable in the first place. Now, in all kinds of research, typically the phenomenon, the things that you're studying, are not part of your everyday life. But everybody has a lot of common experiences, and so there's nothing that I've seen in physics, mathematics, chemistry, biology. There's nothing that can't be explained using models that are found in everyday life. Whether it's a sponge or a car going down the highway or whatever it is, there's always some kind of an analogy that is familiar to the everyday life of a non-specialist audience. It's up to the student to try and come up with what's something that really is similar to the phenomenon they're studying, but is common to everyday life. And so the second rule is to really embrace the complex. And so you need to take your complicated phenomenon and explain it to them using these analogies and not dumb it down. So a lot of people make this common mistake. They just say, there's no way I can take this really complicated phenomenon that took 2 years of undergraduate, or what, for PhD students, it took, several years of PhD on top of their undergraduate to learn this phenomenon. There's absolutely no way I can explain it to somebody that doesn't have some education. But I would challenge anybody to find something that cannot be explained to a lay audience. Everything really can. So the take-home message is, embrace the complex, because if you don't try to explain it, you're dumbing it down, you're talking to your audience like they're children, and then they're just not interested, and that you'll be failing to get the real idea across. All right, so the last one it relates to one of the first things that I talked about, and this is, you don't want to when you're doing a presentation, the idea is to get the audience to understand what it is that you've done or you're doing and how it's important to them. You do not want them to become experts in your field. The more concepts, the more phenomenon that you have to explain, the more they're going to turn off. So the take-home message here is only teach the concepts or phenomenon that are absolutely essential for the audience to understand your research, and nothing else. And lastly, and a corollary to that is, really don't bring the audience down into the weeds. You have to bring the trees up to them, basically. And that's kind of where I'll end for today. If there are any questions, I'll be happy to answer them. Thanks for your attention.
Thank you so much, Dr. Browning. That was an excellent talk. I mean, there are so many situations where I've sat in an audience and I am definitely not the specialist on that topic and have no clue what I'm listening to, and my mind starts to wander. And so that's the whole reason for us doing this toolkit series. So thank you.
My pleasure. So this is available on the CURS webpage?
It will be. It's not just yet.
Okay.
But we will put it up there. I did want to ask whether you have tips that you give your 3MT students for the images that they choose. So, some of these times that we're talking about a real short elevator pitch, you may or may not have any kind of resources to share besides your voice. So if you do have access to slides or whatever, what are your tips for images?
Yeah, first of all, everything that I showed you today can be done verbally as well. And, getting this very complicated image and making and choosing just the key points, I was describing those two experiments, and I could have been describing them without an image. That's for sure. And so But when you do have the opportunity to present something, either with a poster or you're drawing something on a whiteboard or what have it is, then definitely images become important. So if you have the opportunity to do a slideshow or something like that, then, you know, if it's your own research, then it's your own research. But in the 3-minute thesis, we grab images. We have, if you're looking to make cartoons to describe a process or something like that, even, you know, a long slide, there's even posters, there's always a summary that explains your research. So those ones, I really like the students to try and make them themselves, but sometimes somebody's already done a great job. And so for the 3-minute thesis, Google Images, choosing the highest possible resolution you can, and Google is designed in a way to really make it easy for you to do that, to find the optimal images. Meaning you find one that with certain keywords that you like, then you'll see over on the right side, it has that image and then a bunch of other ones that are related to it. And then you can just choose the highest resolution one. Biggest mistake people make is they choose a low resolution image. 400 by 400 DPI is going to look horrible on any screen that you do. The second thing is, and I can give you a link if you want, this will be really useful to the undergraduates for sure. It's called biorender.com. And so this is a company that I found out a few years ago at a cancer meeting, and they were trying to sell subscriptions to it. So you can go there now though, and they have thousands and thousands of images that they've put together, and you just click on the topics. You can put whole things together, and all of the graduate students use them now in their presentations. So you can get pretty high resolution ones to put together. So definitely, if I send you that link, then you can put that maybe on the Careers page. It'll save your students, you know, so much time, because they have mice and you can grab it and put it on your screen. They have a test tube. You can move it and put it on your screen and things like that. So
that's wonderful. That's a great resource.
Yep.
Now, see, for those folks still here, or if you're watching the recording, there is a link in the chat to augusta.edu/curs/toolkit series as one word. And so that's where we'll be posting these.
Okay, I'm going to send my PhDs to this as well. So
yeah, this is a fantastic talk. There's just so many challenges. It's so much harder than it seems to explain something in more simple terms, especially if you have been working on that particular topic.
Well, I can tell you, so we have at least a dozen students in the 3MT every year. We're on year 5 now. On average, it takes me 90 minutes one-on-one with each student just for them to try and figure out what the big impact of their research is and who the beneficiaries are, and then maybe hammer out what could be some key experiments. It's a challenging thing, it really is. But again, I think it's very important, and that's why 3MT has grown so big, because it is important. Yeah.
Agreed. Well, this is fantastic. So we will definitely have a lot of hits on this, and I see Dr. Sadenwasser is saying very helpful, and he'll share it with the thesis students. Certainly, if you're doing a thesis defense, regardless of the level, that's your job, is to narrow down, several years into sometimes 20 minutes, sometimes more or less, but it's not an easy task. So thanks so much for your time.
It was a pleasure. Thanks for the invitation.
Yeah.
Yep. You have a good evening.
Thank you
Dr.
too.
Browning.
Bye.